So let's talk about protein synthesis. Protein is one of the four macromolecules that we've talked about, and synthesis, it means kind of to put things together. So when we talk about photosynthesis, it's the bringing together of different chemicals with energy of light, with light energy, photosynthesis. This is the creation of a protein. We're going to talk about what makes a protein. And we've got this thing that is called the central dogma, and it talks about how DNA will make up, determines what the RNA says, and the RNA determines what protein is made. And so DNA to RNA, RNA to protein. These two things, DNA turning into RNA, that happens in the nucleus, and that process is called transcription. RNA turning into protein, it happens in the cytoplasm, and that process is called translation. So what's actually happening here is your DNA is in your nucleus. RNA is going to do several different things to make a protein, and then that protein is going to determine characteristics of you, so maybe and of living organisms. So that could be determining your eye color, that could be determining your skin tone, it could be influencing your resistance to certain to certain diseases. It could be whether your second toe is bigger than your first toe. It's going to affect the things that we'll talk more about in genetics. And this happens in your cells. So we're going to talk about this story that's happening up here. But first, let's talk about the different characters. We've got three different characters here. We've got mRNA, rRNA, and tRNA. If you'll notice, when we write them, we write lowercase. The first letter is lowercase, and then the RNA is uppercase. I don't have it up here, but if you were to compare DNA and RNA, so DNA, we've talked about before, is deoxyribonucleic acid. It's a mouthful. That's why we call it DNA, not deoxyribonucleic acid. This is going to be very similar to that. It is, instead of deoxyribose nucleic acid, this is ribo. So drop off the deoxy, and you've got RNA, deoxyribonucleic acid. Tomorrow we'll talk more about how the shape of that is different and some similarities and differences with DNA to RNA. And we'll talk a little bit about that today, but we'll focus more on that tomorrow. So this is RNA. Think of it as similar to DNA, but different. RNA, mRNA, let's talk about that. mRNA. I want you to think of messenger. This is a messenger. It's going to bring messages from the DNA that's in the nucleus to the ribosomes, where the proteins, again, we're talking about RNA making proteins. So we've got to start with the DNA. It brings a message from the DNA, and it takes that message to make protein. It ends up making protein. So brings messages from the DNA, which is in the nucleus, to the ribosomes, which is in the cytoplasm. And if we were to draw that, we would draw half of a DNA strand. This is, DNA, we normally draw it where it's a spirally, it's a double helix is the shape that we've talked about. This is just going to be a line with a bunch of base pairs coming off of it. And we'll talk more about that when we get to the story. So the next person we're going to talk about, our next character, is going to be rRNA. I want you to think ribosomal RNA. Ribosomes are where proteins are made, and we've got a picture of it down here, but the rRNA makes up the ribosome. So that one's pretty simple as far as definition is concerned. And then we've got the tRNA, which is probably the more complex. So tRNA, I want you to think transfer or transports. It's got, if you look up here in the picture, we'll talk about this in a sec, but if you look up here in the picture, it transports these things, amino acids. So it is transferring or transporting amino acids around the cell. And again, these three work together to make an amino acid or a polypeptide chain. They work together to make proteins. So it transports amino acids, which I got a little dot here, and then it grows. And as those amino acids are added more and more together, they make a polypeptide. And there's three bases that determine which amino acid it carries. And so the amino acid here is met. We'll see it on a thing in a little bit. I put meth here, honestly, just so if you see this abbreviation, I want you to think not of the drug, the illegal drug, but the amino acid. And then you've got these three letters down here. So let me look right here. It says the three bases determine which amino acid it carries. So the amino acid that's up here can be carried by a specific series of letters down here. So UAC, this entity, this tRNA that I drew here, this tRNA can hold methylamine. These three letters can carry this amino acid. And we'll see in a little bit there's different series of letters can hold different amino acids. And then it can group together to make a polypeptide. So let's get back up here to the story. So we've got the nucleus. Again, the central dogma says that DNA makes RNA and RNA makes proteins. So we're going to start with DNA. And DNA is normally wound together really tight. It's in the form of a double helix. So think of like a corkscrew or a spiral staircase. And then we've got some chemicals. We've got helicase coming into the DNA. And the helicase is breaking and it's unzipping the DNA. And then you've got DNA polymerase, which is not on here. We'll talk about it tomorrow. But it also has a job in here, and it's the end goal here. What we're looking to do is get this DNA code copied over, not copied, that's a poor word, transcribed, which is the word here. We're going to convert it or transcribe it into mRNA. We're going to turn it into mRNA. So you see we got a TAC, and then that would transcribe, going from DNA to RNA. A TAC would turn into a AUG. And you may say, wait, what? We talked about this last time. TAC, during replication of DNA, when it's going from DNA to DNA, it would be a ATC. But here's the thing. That's DNA rules. And when I said this, when I talked to you about the base pair rules, I specified that I clarified that it was for DNA. So A's go with T's and C's go with G's. Apples grow on trees, cars go in garages if you want to. There's other acronyms you can use. But with RNA, we've got something different. C's still go with G's, but A's, instead of there being a T, it's actually a U, uracil. So it's going to be, if you want to use the same kind of thing, apples they fall under the tree. So they start off on the tree in DNA using a T, but now they're going to be a U. So every time you see an A, it's going to be pairing. When we're talking about RNA, the pair is going to be a U. It's not going to be a T. If you ever see a T in any one of these, specifically these mRNAs, if you see that, then something is wrong. There should not be a T. All right, so helicase comes in here. RNA polymerase is going to build this little segment of a gene, and it's always going to be the segment of a mRNA, and it's always going to be in groups of 3. And you remember we talked about that. These 3 bases are what determines what protein is made. So these 3 bases come in, they leave, they exit the nucleus, they come in here to this ribosome. Remember we saw this down here. This red thing looks like a hamburger bun. It is a ribosome. It comes in here. And what's the goal of this, again, is to make a polypeptide chain. And so when it comes to the ribosome, there is a specific tRNA that's in this area that is going to come on and latch on. And if you'll notice, it's hard to see here, but the AUG matches with a UAC. And so that the AUG, this codon is what it's called, these 3 that are on the mRNA, the AUG, these 3 bases will make methionine. It will make that come over here. That amino acid that we call met will come over here. And it will, then the next set of mRNA, this next codon comes in, and then GCU will attract this one over here, that is CGA, and we'll practice this, but the base pairs that match with CGU would be a CGA, and what would happen is this tRNA would go off, the amino acid would pop down, and then the next one will come in and says, hey, you need a CGA to match your GCU, and it will come over here, and the ribosome will use this combination and say, hey, yeah, that's perfect. And then the arginine, ARG, will come over here and it'll add another one. And eventually, like we see over here, it'll add more and more and more to make a polypeptide chain.